All right, well, thank you all for being here. I'm going to be talking about work I did over the summer doing longitudinal modeling of maize gas exchange. So all across the United States, there is an increasing issue with drought tolerance caused by increased climate change. We can see from these graphs that already in the last 5 years, there's drastically increased pressures. And a lot of this is concentrated in areas such as the Corn Belt, where lots of corn is being grown. So this leads to lots of yield loss for farmers. And this is such a prevalent issue that by 2016, 22% of all maize grown was in the US was a so-called drought-tolerant variety. The issue with this being that a lot of these gains have since been attributed to more dense plantings being more normalized in maize growing. This ostensibly leads to more shading of the soil and thus less water loss. So this suggests that these plants were not as suited to drought tolerance as originally thought. And if we truly want to optimize or program a maize genome to have increased water use efficiency, we must look into other mechanisms. So the main mechanism that we're looking at is stomatal conductance. This is basically the rate at which carbon dioxide enters and water vapor exits through the plant's pores. So this process is heavily regulated by SLAC1, the gene SLAC1. And typically stomata are open midday to facilitate photosynthesis, as this leads to more gas exchange and thus more CO2 entering, which drives photosynthesis. So however, at the same time, while CO2 is entering, the water vapor is leaving. And particularly, this is happening during midday, during high periods of high temperature and high solar radiation, which we hypothesize is leading to much more water loss leaving than we would like. So here we use the SLAC1 gene to prototype a programmable plant system where we wish to shift the stomatal conductance to periods of the day with less environmental pressure, such as the morning or the late evening. In this, we compare plants with the wild type allele of SLAC1 with a single knockout, and we'll be doing our analysis based on that and seeing if we can see any favorable phenotypic shifts. So to set up our design, we first started by selecting 8 lines. Here shows the distribution of a whole population of maize, and then we chose across the population to have a representative sample of SLAC1. Because this is relatively exploratory work and seeing the viability of programming SLAC1, we want to be able to look at a diverse genomic population with representatives across the spectrum. Once we chose our 8 lines, we created our programmed mutants by crossing a double knockout to a wild type and selected of the selected lines as shown in the figure to the right here. And then with our individuals, we planted this in a typical randomized field design with multi-reps. So in taking measurements, we were using Lightcore's LI600 barometer tool to take stomatal conductance measurements. This was measuring the rate at which CO2 is entering, water vapor is leaving. We took these measurements over 7 days throughout the season, starting mid-June and lasting through early September. We took 11 to 13 rounds per day, and we sampled from 4 random plants per round. So environmental measurements, relevant environmental data was collected from both the Musgrave Research Weather Station as well as NASA's Power Station. These data were then used to conduct a 2-stage statistical modeling of what the differences in stomatal conductance between genotypes, alleles, and days are. So that's stage 1. Stage 1 is fitting to Gaussian parameters. We do this because we do this to derive Gaussian parameters, ABC. These ABC parameters tell us a lot more information. A is representing the peak stomatal conductance. B is representing the time of day of stomatal conductance. And C is representing the rate at which stomatal conductance declines from the peak. So we calculated this for each individual plant for each day. And the figure to the left is showing the R-squared values or the accuracy values for these. We're generally seeing pretty good accuracy values, except for a few that we believe are due to data points where the corn was starting to die. So these are linked to less quality data points. But generally, we're seeing good quality, which shows that this is probably the correct model to fit to. So with our Gaussian parameters, we then ran stage 2 of the statistical analysis with linear mixed effect modeling. This basically tells us the linear relationships regarding what degree of variation the genotypes, alleles, and environment have in our data. So I use the LME4 package in R to build and forward fit the model. I determined that date, genotype, and allele, as well as the random interactions between date and genotype and the random interactions between date and allele are representing or are capturing the most amount of variation in our data. So then I ran an ANOVA test on the results of our model to determine the importance of the 3 factors relative to each other. So for the sake of time, I am only showing 1 year here, but the general trends mostly follow for both years. So we look at the p-values here. A lower p-value is representing greater significance. And we see that across all 3 parameters, date was highly significant. And for the A parameter, genotype was approaching significance and allele was significant. So in 2024, while genotype was only approaching significant, we did also do the analysis for 2023, and that was significant. So we also see that genotype and allele are, these genotypic effects are only playing a role in the A parameter, or maximum stomall conductance. So we focus on that for the rest of this. So also in so the further results of linear mixed effect models show which of the genotypes are causing the most variation in stomall conductance. Here we look at the estimates of each genotype. The largest positive number and the smallest negative number here represent the genotypes with extreme values, representing extreme variation in the data. So we see that line A661 at the top and line TZUC are the extreme. A661 is the smallest negative number. TZU is the largest positive number. This translates into A661 having the lowest peak, as you can see in the graph here, and TZU having the greatest peak. So in this example, genotype has a very clear effect, indicating that selecting for genotypic background when making efforts to make maize more water efficient are going to be very important. So then now we look at the allele effects. Shown here, the various genotypes for the same day. Shown here are the various genotypes for the same day. And you can see that the difference between the red line and the blue lines are pretty significant, especially for ones like A666 and SD101. And so then we can also see cases where stomall conductance is being shifted in a favorable way according to the difference in the allele. That would be A661 and SD101, where we see the red line is having greater stomall conductance earlier in the day, during times of less temperature, less evaporative effects, less solar radiation. So this is already promising for the shows support for a hypothesis that SLAC1 can be programmed. So now we look at the date. The linear mixed effect modeling was showing this as being the most significant. We look at the particular day. So now we can also look at the particular days that are driving this. And we see, again, at the estimates, the greatest positive value is showing the greatest variation here. And we see that 8-1 this day does clearly have the greatest variation between the plots. So we know that, so we see that environment plays the greatest role as environment is driving the day effects or date effects. So now we want to understand what exactly are these environmental conditions that are driving these shifts between the days, so that hopefully we can isolate the genomic effects from these environmental effects and focus the breeding program on the genomic effects. So to understand what the environmental effects are within the days, and to better understand this, we run random forest regression using the environmental factors as predictor variables for the stomatal conductance values. I used the party package in R to generate many random forest models. I validated them with 5-fold cross-validation and chose the most accurate model listed at the bottom for each of these with their quality scores as well. So all these are about 0.6 R squared, which are pretty good quality. So from this, we see that for both alleles, the most important environmental factors are air temperature maximum and the average air temperature, as well as relative humidity mean. We also see that solar radiation and wind speed are showing up as top environmental factors. So with this work, we were able to characterize a lot of the differences from the SLAC1 mutant, as well as some of the differences between the genotypes. We showed that the environment is a major driver and that stomall conductance is a very complex trait, and that it's not going to be able to be influenced by just mutating an allele or something like that. This suggests that a breeding program using SLAC1 would likely be focused on breeding for the quantitative trait of drought tolerance, and that we would need to optimize both the genotypic background as well as the SLAC effects. We did also look at double mutants of the SLAC1. In this presentation, I mostly talked about the single mutants that I analyzed over the summer, but we did also look at double mutants, and those were having a greater effect on the phenotype than our single mutants were here. And that is described in our manuscript as well. So yeah. So, and then I'd like to call out Sam Mosier and Harold Backer for leading this project in particular. And thank you.
Questions for Nicholas?
So, you mentioned that, like, at the beginning, you chose like a variety of plants.
Yeah.
Regardless of population, right?
Yes.
So, I guess, like, given that you chose a wide variety of plants, do you have any idea of like the specifics of like the genetic background that might be influencing like the
Yeah, I'm sure it's probably in literature, but we didn't really look too much into what exactly are in those different lines that's leading to the differences. I believe it is mostly just to do with the level of expression of SLAC1. So so I think to answer your question, it would mostly just be how much of the SLAC1 protein is being generated and then being involved in stomall conductance down the line.
Is there a trade-off, do you think, with stomatal conductance and other traits like photosynthetic efficiency or maybe like heat tolerance?
This definitely is a big thing in the area of trying to increase the water use efficiency in maize. You know, I know one common thing that's been used is a lot of people will just remove the pores from the plant, and then obviously you can't get you can't get CO2 in, so that really doesn't work. So that's actually why we were trying to modulate SLAC1, so that hopefully if we can get it, you know, being more open, greater rates of stomall conductance earlier in the day, that leads to less water loss from like evaporative effects. But it's not like there's any less CO2 in the atmosphere during the day. So hopefully that is negating the trade-off. But we do also have to keep in mind that like once we are changing the SLAC, we are seeing there's shifts in the peak. So maybe that would be the trade-off. But we are trying to eliminate that trade-off by programming SLAC1.
What's been the most challenging part of this work for you, and what's been the most rewarding?
Yeah, the most challenging is definitely trying to navigate how the different environmental effects are impacting the different genomic effects. We started this really being interested in the differences between SLAC1, the mutants and the wild types. And a lot of that is clouded by the environment effects across the season, and even within like the day, various hours. So that's been really hard to try to isolate. Most rewarding, I'd have to say just trying to make sense of this data. This is pretty exploratory work, and I think it'll have some real ramifications for farmers in the future.
Thanks so much.